killing efficiency, this graph that I showed you where we were killing things at a decimal reduction time of 1 minute is typically from an autoclave at 121 degrees Celsius. You can calculate the decimal reduction time. Another thing that you could do is calculate the thermal death time, which is the shortest time needed to kill all microbes. The issue with this one is if you start with more microbes, it can take longer. So the decimal reduction time is more commonly used. Okay. So in this particular case, looking at this graph, what is the thermal death time? The shortest time needed to kill all the microbes? 7 minutes, right? Okay.
Are we going to have graphs for this?
Oh, yes. There'll be the graphs where you have to figure out, like, is it alkalophilic or acidophilic, like those, or acid tolerant versus the graphs like that. And there might be graphs like this where you would calculate the decimal reduction. And I think there's a homework question as well to help you practice that here. Yeah? All right. Sometimes you don't have access to an autoclave or you can't autoclave something, and you may use heat to do pasteurization. So pasteurization is a process of controlled heating and cooling where you, and it's typically only done with liquids because you have to kind of force them through tubes that move them between the temperatures. What are some examples of things that you come into contact with that are pasteurized? Milk is the big one, right? Which is why, and is pasteurization bacteriostatic or bactericidal? It's static, right? Because if you wait too long, then the microbes can still grow after you pasteurize. Yogurt actually, yeah, and yogurt has live cultures. So, yeah. So it's kind of like always growing. But yeah, so yogurt can be pasteurized. What are some other things that can be pasteurized? You can't pasteurize wine. Yep. Juices or cider is another common one that can be pasteurized. And I think any like semi-solids like yogurt can also be pasteurized. Okay. So you're heating and cooling. You can do beer as well. And again, this is not sterilization, but really it's a form of sanitizing, right? We're reducing the microbes to an acceptable level, but because after we stop pasteurizing, we know that the longer we wait, the microbes can potentially grow and come back. Makes sense? You can't, it doesn't have to be, but that's one of the issues. So like every year, there's like a tragic case of somebody who gets like E. coli or salmonella from apple cider where they've made it like at the farm, and if they use apples that have gotten into contact with animal feces, then you can, and it's not pasteurized, you can then develop diseases. Whereas if you but most small farms don't have the capacity necessarily to run pasteurization, right? But most of the cider and juice, if you get it at like a grocery store, it's a bigger operation, they often will pasteurize. So it doesn't have to be, just kind of, but there is risk involved if it's not pasteurized. You know, like, so it's not as good.
I find it interesting. I want to talk to you about that.
I talk about it.
Oh, yeah.
I'm like, oh man, it's fall. We're going to have trouble again. All right, so let's talk about, so again, dry heat sterilization. So here we have a researcher. She is inoculating a tube of media, and she's got a loop. What do you guys do to sterilize your loops in the lab? We burn them in a Bunsen burner, right? There are also these dry heat incinerators that will heat them up as well. But that's the idea, right? You are killing all the microbes that are present. It's non-destructive in that case. Why? What's the loop made of? Metal. So it can be heated to very high temperatures without being destroyed or melted. Yeah. We also sometimes have plastic loops. Can you put those in a Bunsen burner? No. Those are considered disposable. All right. So when might you use dry heat incineration? Would you dry heat incinerate a bottle of media? No, because you burn and boil off the media, right? But what if you were trying to sterilize like a powder? Let's say you were going to have a powdered medicine that's a solid. What would happen if you put it in an autoclave? That steam's going to get it moist and clumpy. So if you need stuff to stay dry, it may make sense to use dry heat incineration. This can be also used with electronics if they're not going to melt. Glassware can be incinerated. Powders and oils, you may put them in an oven that gets to very high temperatures. If you have anything that is moisture sensitive, you might use dry heat rather than moist heat.
What's the most common use of dry heat?
Oh, probably waste. Yeah, great question. I'd have to look it up to be sure. All right. What's going on in all of these cases? Why does heat work to kill organisms? Do you guys know? Yeah, they can survive. Yeah. So in part, I mean, if you burn something, yeah, I mean, you've just destroyed everything. But like thinking about like, why does heating to close to boiling work for some things? What happens to your proteins at high temperatures? They denature, which means they lose their structure, which means they lose their, say it again, structure. Structure. Structure, structure, structure. Good. Okay. So, and this is the same, right? So what's happening with dry heat, if you're actually incinerating, you destroy everything, basically. But at high temperatures, either in the autoclave, you are denaturing proteins and disrupting membranes in many cases. And both of those are very bad for living cells. Let's talk about UV radiation. So we use UV radiation in the laboratory all the time, but the key with UV radiation is that it is a surface level disinfectant. You are already familiar with the fact that UV radiation can cause damage. Where do you run into UV radiation? From the sun. And what do you do to protect yourself from the sun? Sunscreen or, yeah, clothes or a hat, right? Because can UV light penetrate your clothing or even through the sunscreen? No, it can only go to the surface. So even when we talk about you can use UV light to sterilize water, but you have to make sure it's a very thin layer because it's only going to penetrate a little bit. Okay. The reason that UV radiation works against living organisms is because living organisms, including viruses that are not living, I'll add viruses, have nucleic acids. And UV radiation causes a specific type of mutation, right? This is one of the reasons that we warn you from getting too much sun, especially if you look like me, yes? We're the people who look, oh my God. I can't go outside like at all between 10 and 2. It's like a no-go zone. I have like the world's biggest hats. I have like bathing suits that are long sleeved and I'm like, if I go out, I will burn. I look like this, and then I turn bright red, and then I go right back. Mm-hmm. Yeah, like we can play this game of like those of us who are pasty. Okay, so We've got thymidine dimers, which then mean that your DNA is changing its structure, which changes its function. If these happen in anything that is critical, that means that, right, how do we use DNA in a cell? It's the instructions for making what? How do we use DNA in a cell? It gives us the instructions to make what type of macromolecule? Proteins. And proteins do?
Everything.
It's like you're paying attention. Good job. Okay, so if we've destroyed the instructions for how to make proteins that do everything, that's bad, yes? That is bad. Okay, good. So we can use this in this case. What are we trying to sterilize here? Well, not the objects, just the surface, right? So we're basically trying to sterilize like this working surface and hopefully all this other stuff that's in there, like the surface, but not what's inside, because if it's inside, it's protected from the UV. If we really want to get, yes, Tyler. Which things? So like these, if this is like tubes, only like the outside of the tubes would be sterile, not the inside.
Well, what about underneath, like, where those are?
Nope. That's why I like to keep my hood, like get everything out so that it is like all the surfaces can be sterilized by UV. So, but if you want to have more penetration with your radiation, you can change to what's called ionizing radiation. So here we're talking about X-rays and gamma rays. Talk about sterilization. Awesome. All right. So what do you guys know about X-rays? Can X-rays penetrate inside things? Yes, because what do we use them for? Right. We can expose your body to them in order to see differential density, right? So we can see bones because your bones are denser than your soft tissues. What happens when you go get an X-ray? What if they're doing like an X-ray of your mouth? What do they put on you? A lead vest that covers what? All the other important bits, right? We're really worried about your organs that you need, your lungs, your heart, all that, and your gonads. So the organs that would produce your gametes, right? Because we don't want to have any mutations, because X-rays can also cause mutations. They can cause double-stranded DNA breaks, which is bad, bad, bad. So, and what happens to the technician who's administering the X-ray? She like goes outside the room. Like, this again, they're like, I don't want to be in here because X-rays can damage DNA. Gamma rays are even more penetrating. A lead vest would not stop a gamma ray. Okay. But the beauty of that is, so if I expose these plastic tubes, does the plastic give a stop? The X-ray or the gamma ray? No, right? It's going to go like in, through, out. So this is how, I'm going to talk about, so lots of the things that you interact with that come wrapped in like paper, like bandages, gauze pads, the tubes that we use in the lab, they are sterilized by ionizing radiation because what we're able to do is package them in a non-sterile way, and then we put them on a conveyor belt, and it carries them automatically down past a radiation source, and then can that radiation penetrate the packaging? Yup. And then they come back out. Why do we not want to send the workers in there? That would be bad. So it's got to be automated, and then they come back out. But yeah, almost all the stuff that you guys work with in a clinical setting where you have to like unwrap it. Tell me the other things. Like, what do you like? Open packaging. where you like have the two, like bandages, I know, gauze pads, like almost all the like plastic syringes, like keep going. What else? Oh yeah, the tongue depressors. Yep. All of that is, yeah. Yeah. And again, that's, you know, if your gloves are sterile and come, yeah, then they probably, yeah, packaged. That's probably. They like, you know, fold open the extra. Right. And, like, not, the whole thing. My gloves are not sterile. I just pull them on and then spritz myself with ethanol because it's a disinfectant. Okay. Did you know? Did you know that some of your food gets ionized radiation? I know you're like, this is bad. Right, does it stay in there? No, you don't worry about ionizing radiation on your bandages. But here we can see that if you radiate strawberries, look how before getting moldy, because what have you done? You've killed off the spores and the infectious material, which lets it then last longer. Well, that's a problem because we don't want to let you do it at home, right? But yes. People are concerned, right? Do you want to live next door to this facility? Probably not. Because again, the radiation goes through, has done its job, and it's no longer there, right? Again, you put gamma irradiated bandages on all the time and you don't worry. This is what I'm saying. People get weird about food. I don't know why. It's okay. That's okay. All right. So, as a reminder, you guys are going to look at mechanical removal methods on your own. Basically, it's filtration. And again, what happens if I try to force a pair of gloves through a filter? Nothing. So you can only use it for gases and liquids. All right. Let's talk about switch gear.